Hi, this is Jongyun Kim from University of Georgia, and this presentation was presented at SN Research Conference in 2011. And this study was about how the Petunia plant acclimated at different substrate water contents. Plants feel thirsty when they have drought conditions, and they have lots of responses to this drought, such as reducing growth, closing stomata, reducing photosynthesis, and the other responses can be happened by this drought. But this drought physiology can be used as a strategy. Imposing mild drought can be used as a strategy in nursery production. It can slow plant growth and they can make better marketing time. And sometimes they have post-production benefits by hardening of the plants. This kind of mild drought can make longer shelf life or it can give the plants with better landscape performances. And this kind of strategy is by the acclimation factor of the plants. But we have a question here, how mild drought is actual mild drought? Because this is ambiguous, we try to look at the substrate water content specific response, how they acclimated in physiological responses or plant growth, and which is the beneficial level of substrate water content which can acclimate better and give a good plant quality. We use Petunia apple blossom cultivar as our plant. We planted 8 plants in a liter tray, and we use Fuffer Tupi mix as our medium, which is peat moss and perlite based. And we had the irrigation system with drip irrigation with customized grid, shown at the picture. And we had 4 treatments by 10, 20, 30, 40% substrate water content. This is the irrigation system. The soil moisture sensor EC5 from Decagon device is inserted in the substrate, and it sends the substrate water contents by this data logger. And if the substrate water content is less than the set point, it opens the solenoid valve and it waters the plant. And this system could maintain the substrate water contents constantly throughout experiment. This is the picture how we grew the petunia in the greenhouse at UGA. We had leaf physiological measurement with Cyrus-2 system. It could measure leaf photosynthesis, stomata conductance, transpiration rate, and chlorophyll fluorescence. We measured every 2 or 3 days at noon time. So from this time, we could compare between when they reached the set point, substrate water content, and a week after, so we could look at their acclimation abilities. We measured the uppermost fully expanded leaf size at harvest and leaf relative water content. We used randomized complete block design with 4 treatments with 4 blocks, and we used SAS to analyze the data, and we used analysis of variance, and we used Fisher's protected LSD at alpha is 0.05. This graph shows the substrate water content change by time, and this arrow indicates when we measured their leaf physiological responses, and these closed symbols showing during the drying period, and these open symbols showing that after they reach the set point. So as their physiological responses, on the left is stomata conductance and right photosynthesis. On the x-axis is substrate water content, so as substrate water contents decreased, the stomata conductance and photosynthesis decreased during their drying period. But after they reached the set point, they increased back in 20% and 30% treatments, which shows their acclimation of the plant. So to compare when they reach the set point and a week after, we try to compare these 2 points. So this gray bar indicates when they reach the set point, and it is just the same as the graph that we have seen just before. But after a week period, they have some changes in their stomata conductance, but they have no big difference, but only 30% have significant increase of stomata conductance after a week, and 10% has no difference at all, and it still maintained very low stomata conductance. Also, the transpiration they had changed, but only 30% has significant increase in their transpirational rate, and also 10% they had no change at all, with very low transpirational rate. From chlorophyll fluorescence measurement, we calculated quantum yield of photosystem 2. It shows how the plants use the light in photosynthesis, and they had no big difference between when they reached the set point and a week after, but they had only the treatment differences, and 10% had the lowest quantum yield of photosystem 2. But interestingly, in photosynthesis, as you see here as mean separations, 20% and 30% significantly increase their photosynthesis after a week period, and it shows the acclimation of the plants, and 10% had no difference in photosynthesis, even they seems like they decreased their photosynthesis. At harvest, the leaf size of the plant showed the typical drought responses. The lowest substrate water content plant had the smallest leaf size, and 40% substrate water content plant had the biggest leaf size. But interestingly, the leaf relative water contents, only 10% plant had the lowest relative water contents, significantly lower than 20 or 30 or 40% substrate water contents. It showed kind of wilted leaves at this point, which shows very severe drought condition at 10% substrate water contents. So in conclusion, we have looked at the substrate water content specific physiology of the plant, Petunia apple blossom, and we looked at their acclimational responses by their drought at the different substrate water contents, and we figured it out the severe drought could happen at less than 10% substrate water contents, which has no acclimation in photosynthesis or anything at all. But we could say that the substrate water contents from 20 to 30%, they had a good acclimation and they were beneficial, and it may be used as a good strategy in nursery production to make a slow plant growth or better performances for landscape as a post-production. So we use this kind of references, and I'd like to thank the Sudo, our technician at the lab, and Decagon Device for the soil moisture sensors, and USDA for funding this research, and Farfart for donating their substrate. And if you have any questions, please email me, Jongyun@uj.edu. And thank you for listening to this presentation.